Okay, welcome to podcast 23. This podcast is dealing with the nervous system, and in particular the function of the neuron, which is a nerve cell, which is the fundamental unit, functional unit of the nervous system. This is nested inside the overall cell communication category. In your podcast, the immune system is in this group, the nervous system, and the endocrine system will be also. So just a reminder, if you were to see an essay question on cell communication, this is another system that ought to come to mind right away. In order to send nerve impulses through your body, cells must communicate with each other. Nerve cells must communicate with each other, and they must communicate with other body cells, like muscle cells, for example, to communicate to them to do something, like contract. No, I do not. Why would you ask me that? Okay. At any rate, so let's get started. All right, so focus on the neuron and the transmission of the nerve impulse. Now, why do animals need a nervous system? Okay, animals are doing stuff like catching prey items, trying to evade being caught. Animals are active in ways that, for example, plants are not, and to respond to that environment in an immediate way, a nervous system is necessary and obviously quite helpful. Now, what do animals need in a nervous system? Well, if you are a hawk going after a bunny, poor bunny got caught, you want to be fast. You want this system to be accurate, and very important to what we're going to be talking about, you want it to be reset quickly. Once a neuron, for example, has fired to help you catch the bunny, or if you're the bunny, maybe to help you evade being caught, you want to be able to set up that nervous system again so that you can continually respond to stimulus, and you're not it's not just one time and you're done. So neurons meet all of those qualifications. Neurons fire signals very, very fast. We can set them up quickly. It's very accurate. So let's talk about this fundamental unit of the nervous system, the neuron, a nerve cell. It's just another word for a nerve cell. Okay, neurons are really unique looking cells. When we think of cells, when we're first taught about cell structures, we think of these, globular things with a nucleus in the middle. Okay. Well, if you look at this photo electron micrograph of a neuron here, you can see that it is far from being just a spherical shape. It is very long with many arms stretching off from it in all directions here, and then this one main trunk coming off of this central body right there. They have very characteristic shape, many branches from them, and that's very pivotal in how they work. Okay, their structure is related to their function. This idea of structure being related to function is something we've been trying to get at all year with different cellular structures, tissue structures, and here's another example. In this case, we've got many places where nervous signals could be received by this neuron. Sensory information can be inputted into the neuron, and this one main trunk where this information is going to be sent to some target cell, perhaps a muscle, so that you can respond to that stimulus. We'll talk more about this as we go. Okay? All right. The neuron structure, I think in your handout you're asked to draw and label this, so this is really, really important that fundamentally to everything we do that you understand the parts of the neuron, so make sure you get this. First of all are the dendrites. These are the many, many branches that come off of the central cell body. You can see that the cell body contains the nucleus and ER and all the other parts we normally associate with cells. The dendrites branch off of that cell body. Okay, this is the axon, this long single branch. It looks kind of like a overlong dendrite, but this is the part that is going to send the nerve signal on to perhaps another cell. So that's the axon. You can see that the axon, it looks like it's got like 15 or so little rice grains here. Well, they're not rice grains. They are cells called Schwann cells that are made up of a fatty type substance called myelin. Collectively, this whole thing is called the myelin sheath, and that myelin acts as insulation, so to speak, and it also has another purpose that we'll talk about later. But when you put wiring in a house, you don't just put a bunch of bare copper wires together. You insulate those wires with plastic or rubber so that they don't interfere with each other and they don't short each other out. Well, you need to think of the myelin sheath as like that. It's going to protect the signal as it goes through this axon. Okay? At the end of the axon is the terminal, or the end of the axon. This refers to it as a synaptic terminal. Mainly, you need to understand that's the end of the axon right there. Okay, and at the end of the axon, there is empty space. Normally, on the other side of this empty space, there might be some other cell. Okay, perhaps a muscle cell or another neuron over here for the signal to jump over to the next neuron and be transmitted through the body. But what you need to understand is that there is a space where there is fluid, perhaps, but nothing else. It's called a synapse. That's very important, and we'll talk about how the role the synapse plays in nerve impulses later on. Okay. All right. Now, the signal direction. The nerve impulse always travels from the dendrites through the axon and towards the synapse. That's very important. So this is the direction the nerve impulse travels. It always goes in that direction. Very, very important that you get that in your head. That's the way we're going. So dendrites to cell body to axon to synapse. Okay? All right. Just some interesting facts about neurons. Neurons make up some of the longest cells in your body. A blue whale neuron could be up to 30 meters long. They've been measured. So extremely long cells. We don't normally think of cells as being long like that, but a giraffe axon measured at 5 meters, very long. And even in your body and your leg, that's where your longest neurons would be, 1 to 2 meters, depending on your height, going down from your lower back into your leg. So the nervous system allows for an incredibly fast response time, a millisecond response time. So extremely fast and kind of unusual in their physical structure as compared to other cells as well. Okay? All right, as we talk about how nerve signals are passed through a neuron. Now, right now we're talking about just through a single neuron. A single nerve cell receives a stimulus, and that stimulus is going to travel through the neuron to be sent somewhere else. You need to think of dominoes, okay? When you start the signal, for example, when you look at these dominoes down here, to start them falling, you just push the first one and the rest fall, okay? You tip the first one, and that is the trigger of the entire signal, okay? The signal propagates as the dominoes fall against each other. Please note, the dominoes themselves do not pick up and move to the end of the line. They just send this wave of energy through the line. Okay? And then if we want to do the domino trick again, we have to reset them up. That is also how it is with a nerve cell. If you want to fire it again, you have to reset the system. Okay? You have to set the dominoes up again. You have to reset the neuron to make sure it's ready to go again. Okay? So, in a neuron, I want you to think of the dominoes, so to speak, as being protein channels embedded in the cell membrane of the neuron. We are very important that you keep in mind the importance of cell membranes. We've talked about it with the immune system and the identification markers on their surfaces. We'll hear, and in the nephron with all the channels, we're going to be focusing on channels again. But this time it's not going to be water that's the main thing that's flowing through the channels. This time it's going to be other molecules, ions, that are moving through these channels. But there are channels embedded all through here, and they act like dominoes. If this is the first channel right there in our domino chain, and we do something to it, then that event is going to be passed through all of the channels as they go down through the neuron. So those little protein channels will act like dominoes. If something happens to the first one, it gets tipped, the others will get tipped in turn. Okay? In order for them to work, therefore, they have to be set up, and when they get tipped over, they have to get reset if that neuron is ever going to fire again. Okay? So, first channel is open. Once the first channel, remember we open and close channels to allow things to pass. Once the first channel is opened, the rest will open after them in order along the axon. Okay? Now, this is an all or nothing type of response, meaning if we're going to make the dominoes fall here, we have to have enough oomph, enough energy, enough stimulus, if you will, to push down the first one. If there's not enough energy to push down the first one, then none of them are going to fall. That is the same with these little channels embedded in the cell membrane. If there is not enough of a stimulus to get that first one to open, then none of the rest will open. Okay? It's important. Now, I know you're like, opening for what? Well, we'll talk about that in just a minute. But understand, it's all or nothing. If we don't have enough stimulus to activate the first channel, none of the rest will open. But if we do, a wave is going to pass through all along the length of the neuron. And then we will have to reset the channels to be like they were in the beginning in order to do the impulse again. All right. Ions. Ions are really, really important in how neurons function. Okay? So this is a neuron. Consider this the axon of a neuron. So it's a long part of the cell. This is the cell membrane right here, and this is the cytoplasm of the cell inside. So all of this is membrane. All right, the membrane is what we're going to kind of focus on because that's how things get in and out. But what things? So let's talk about this. Neurons, like all cells in your body, live amongst a fluid, and in that fluid is a bunch of charged particles, among other things, but we're focusing on the ions, the charged particles right now. There are anions, negatively charged particles, and these anions tend to be more concentrated inside cells than they are outside of cells. Okay? There's lots of, as you can see here, chlorine ions that are negative, and there are some outside the cell, but there's lots more inside the cell. That's not trivial. That's important because remember, this sets up a concentration gradient. Where is the concentration of chlorine high? Well, it is high inside the cell. Where is it low? Outside. Okay? I want you to keep thinking about that idea of a concentration gradient. Okay? There's all kinds of proteins and amino acids, and generally speaking, amino acids and proteins tend to have some negative charge about them. So these blue blobs here, these are proteins, and you should consider them negatively charged. Amino acids, which as you might recall are the building blocks of proteins, they are also negatively charged, and they tend to be in higher concentration inside the neuron than outside. There's not very many of them out here. In fact, I didn't put any. So high concentration of negative in, low concentration of negative out. Now, there are also cations present, positively charged ions. Now, these cations tend to be more concentrated on the outside of the cell rather than inside, okay? So, there's lots of sodium ions on the outside in the extracellular or outside body fluids, outside of the cell's cytoplasm. It's not that there might not be a sodium ion in here, there could be, but there's going to be more of them outside. Okay, so the concentration of sodium is high on the outside and low on the inside. Okay, also, however, there is another little caveat we need to mention. There are also potassium ions, and those happen to be, even though they are positive, they happen to be more concentrated on the inside of the cell than on the outside of the cell. Okay? Now, even though there are potassium ions in here and they have a positive charge, please note there are still way more negatives on the inside than there are positives. So overall, this neuron is going to have overall a negative charge on the outside and a more positive charge existing. What did I say? Did I say inside? Inside, and a more positive charge existing on the outside. Okay? That's the bottom line you need to take away from this. So, there is a charge difference from outside of the cell to the inside of the cell, the neuron, and it looks like this. Okay? It is more positive on the outside and more negative on the inside. Okay? And here's your cell membrane right here. Now, you could think of this as a concentration gradient or a charge gradient, okay? There's more positives out here, more negatives in here, and it's a gradient. If the positives could get in, they would. They would flow with their gradient. If the negatives could get out, they would. They'd flow with their concentration gradient, okay? It's important that you get this concept. More positive on the outside due to the presence of lots of sodium on the outside, more negative on the inside due to the presence of lots of anions, chlorine, amino acids, proteins, all of which have negative charge, okay, associated with them. Okay? So, because there's more negative on the inside, more positive on the outside, these neurons, and cells in general, but particularly these neurons, have voltage, okay? Opposite charges on opposite sides of the membrane. There is electricity here, is the bottom line. You are an electrical being. We don't think of it like that, but we are. And it is certainly in no small part due to this voltage difference across the membranes of your cells, okay? When this cell is set up like this in its normal state with the positive and higher amounts on the outside and the negative higher on the inside, we say it is polarized. Remember what polarity means. You have two different ends, two different sides, okay? That is being polar, okay? Well, this membrane is polarized. And boy howdy, if they could, these positives would move this way, these negatives would move that way with their concentration gradient, and the thing would become unpolarized. But they can't because the cell membrane is not allowing it. Okay? So, again, they would reach equilibrium if they could, but they can't because that cell membrane is semi-permeable and it's not allowing that movement. Okay. So you can think of this as a charge gradient, like a concentration gradient, and you need to think of this as being stored or potential energy. When you've got stuff that wants to move, but it can't move, that is stored energy there, just waiting for the dam to burst, okay? All right. So, how much voltage is actually existing between the outside and the inside? Can you measure it? Well, they can. You have very evidently tiny little voltmeters that can measure this kind of thing. And in a normal neuron, a normal resting, not firing, not doing anything neuron, the voltage difference between the inside and the outside is a negative 70 millivolts. So it's quite a bit more negative on the inside than it is positive on the outside. Okay, so a negative 70 millivolts. Okay, this voltage of negative 70 millivolts is called the resting potential. Okay, this is the resting potential, meaning the neuron is at rest, it's not doing anything, and if it were allowed, the charge difference here would reach an equilibrium, which would be zero, with no difference between the outside and the inside. But it's not being allowed. These charges are not being allowed to move because the cell membrane is preventing them. Okay, so it's a negative 70 voltage between the outside and the inside. Okay. All right. Now, protein channels are going to be really important in controlling how these ions move and allowing an electrical impulse to flow down the neuron. Okay? Remember that there are all kinds of protein channels for different types of molecules in different types of cell membranes. Not all cells have the same kinds of channels. That is what makes certain cells function in different ways. The cells in your nephrons had lots of aquaporin in them so that water could flow out of those nephrons and be stored in the body and not lost with the nitrogenous waste. Well, in neurons, I'm sure there's some aquaporin, but that's not the focus. The focus is on different kinds of membrane transport proteins that allow other kinds of molecules to go back and forth, because now our focus is on electricity and sending an impulse through the neuron. Okay? So, just remember that part of what makes cells different from each other is that they have different proteins in their cell membranes that allow them to do different things. It just makes sense. Okay? All right. How does a nerve impulse travel? First thing that has to happen is it has to get started. We have to tip over the first domino. Okay? So, a stimulus, something, kitchy kitchy coo, you get tickled, whatever. A nerve is stimulated. Okay? But remember, that stimulus has to be significant enough to tip over the first domino. That means we call that meeting a threshold potential. If there is a stimulus, but it is not strong enough to tip over the first domino, then you're not going to have a nerve impulse. Okay? So, what we mean here by the first domino is a sodium ion channel, a protein channel that allows sodium ions to pass. In its normal resting state, these channels are closed. When there is enough stimulus, however, a sodium ion channel will open and thus allow sodiums that are in high concentration on the outside of the cell to flow inward. So, if we we tickle the cell and we tickle that sodium channel that might be right here and we cause it to open up, okay, we open it up. That will allow the sodiums that were in high concentration out here to begin to flow in. And they will flow in to such a great extent that, in fact, the polarity of the neuron, this little section of the neuron near that channel, will flip-flop. Okay? We're going to let in so many positives that it's going to make it positive on the inside, as you can see has happened right there. Okay? This is really important in moving the nerve impulse down the neuron. Okay? Now, if the stimulus was not strong enough, then nothing will happen. But if it was strong enough to open that first channel, then those sodium ions, and please note what process this is. It is diffusion. They are going to flow with their concentration gradient, or voltage gradient if you prefer, because we're really focusing on the electrical part of this now. But it's molecules with charge. That's all it is. Okay? So, at that specific point on the neuron, the charge is flip-flopped. Instead of being more positive outside and more negative inside, we let in so many sodiums that we increase it to a very positive charge right in here, and it's relatively negative on the outside in comparison. Okay? We say here that now we are depolarizing this neuron. It is not polarized anymore. It is depolarized. Okay? The first domino went down. All right. Now, the thing is, we want this wave of impulse to move in this direction towards the end of the axon. We don't want it to go backwards, back towards the dendrites, back towards where our stimulus probably came from in the first place. So how do we keep, when the sodiums first rush in, okay, we want to keep the impulse going this way. How do we make sure it doesn't go that way? Well, here's what happens. When the sodiums came in initially, okay, that charge change that occurs as we make this more positive on the inside and negative on the outside, that flip-flop of charge that occurs causes this channel that might be here, that remember there are channels, sodium channels all along here. When we flip-flop the charge here, that stimulates this channel to open. And so sodium rushes in and we flip-flop the charge. Well, that stimulates this channel to open. That was interesting. And we flip-flop the charge. And that stimulates this channel to open, and we flip-flop the charge. So once you flip the first domino, all the rest will be stimulated to fall. How do we prevent it from flowing this way? Why doesn't it flow backwards? Well, here's why. There is another type of channel at work. Okay? In order to make sure we don't flow backwards, we're going to have potassium channels also be stimulated to open. Okay? These potassium channels, because of the opening of the sodiums, will open up right behind them. They are also embedded all along this neuron, these potassium channels, K+ channels. Okay? Now, what will happen is that these positive potassium ions will flow. Remember, we had potassiums in here in high concentration. They will flow with their concentration gradient out. So we've got sodium coming in, but right behind the sodium that's coming in, we've got potassium going out. What that does is it restores this to being negative and this to being positive, okay? Inside negative, outside positive. Now, that's important. Why? Because as long as we restore to this state, none of the sodium channels at this end of the neuron will be stimulated to open. So we keep our flow going this way, okay? So we're resetting the voltage gradient, so it is like it was when it started, and we're going to keep the impulse going in the direction we want. And we restore the positive to the outside, the negative to the inside. Okay? And we've already talked about that. All right? Now, okay. I think I should stop there because I suspect that you're pretty bored of this. I think I'll stop there and we'll finish up with a part B later on. All right? Thanks for watching. I'll catch you on the next time. See you later.